from INRA. INRA is a National Research Institute for, the name is agriculture, but we have three aims. Agriculture, of course, but for environmental issues. Agriculture is for producing food and producing non-food materials, so it's not necessarily food. But all food and non-food has, of course, environmental impacts. So we are at INRA, we have these three targets, environment, food, of course, and agriculture, of course. And all three are links because this is obvious, and it's very difficult to work on one topic without any looking at what happened for environmental concern or impacts, and what about for where the food or the materials come from, and so on. So here I just want to go on a very focused topic. It's here it concerns the removal of bacteria and specific form of bacteria, which is spores, and from stainless steel. Why stainless steel? Because stainless steel is widely used to in food processing equipment. And here I will show some data on results concerning data on cleaning in place results. And if we look at the objectives of this work and why this work, and first of all, you have detailed different elements, or given different elements about bacteria, and of course bacteria. We can't speak about biofilms in the gut because bacteria are in they are not only bacteria on the wall of the intestine. Sorry, I don't have the vocabulary, but they are bacteria inside the material you have in your body. And here we look at bacteria that attach onto surfaces, which is a normal situation for bacteria to be attached to a surface. And then they scarcely remain as just attached, and they produce biofilms. So spores, it's not vegetative cells. So it means when you speak about spores, you speak about dormant bacteria, dormant form of this bacteria. So why I will explain, I will give you why there is a huge link in between biofilms and bacteria spores. So we will look at that through some examples. But in fact, we have vegetative cells and spores that could contaminate food products. And this afternoon we did have some examples about milk, and we did have a look on the effect on some techniques to kill this bacteria. And we found that, for example, the first presentation on pulse electric field, sorry, was ineffective on spores. So if I have properly understood your presentation. So if we go if we remain at spores, we know that spores are highly adherent on equipment surfaces, especially on stainless steel, but not only on stainless steel. And they are resistant to heat, resistant to chemicals, and resistant to flow shear stress. What it means, it's resistant to mechanical action when you want to remove by flowing just the shear stress due to the flow on the surface. I will show you some data on that. It's very difficult to remove these bacteria from the surface by just flowing, just the shear stress due to the flow on the surface. So what happened for sanitation in production line? In fact, when we clean, all this surface, the objective is to eliminate most of the soil, and not only organic soils, but everything. And the less bacteria we have on the surface, the lower is the risk of recontamination of food. Of course, you can say that why not using, because in the food industry, you have cleaning, of course, but you have disinfection. But if it's well known that if there are disinfection, you lower the population by a certain amount of power of 10. So the more bacteria you have on your surface after cleaning, the less effective could be your disinfection process action. So for industrial point of view, cleaning appears to be the most important part of the hygiene process. The most bacteria you remove, the easier is to maintain your food safety at a correct, acceptable level. So here I will show you some data about the removal kinetics of spores from different bacillus strains. And we did look at the roles of chemical and mechanical actions in the cleaning process. And I will end my presentation by some related issues that could be considered to be important for application and for industrials. So let's start by the description of material equipment. We work in the lab. Most of the work is done at a pilot plant scale, but we have some lab scale experiments using what we call laminar flow cells. It's something where you can flow your detergent above a surface and have a look through a microscope. So it's a widely used technique at a lab scale, and it's possible to visualize the removal of your contamination through the microscope. But for industrial application, it's difficult to convince industrials based only on this kind of experiments, because the scale is not enough to have all the phenomena that could explain what happened in the real life. So you are obliged to work on a higher scale in order to have all the phenomena involved in the cleaning process. However, it's important to work at these two different scales in order to understand the mechanisms involved. So, for example, here we worked with stainless steel pipes, which is widely used in the food industry, in the food processing lines, and we worked with cleaning cleaning conditions which are really could be considered as mild, because we worked with 0.8% of sodium hydroxide, which is widely used, for example, in dairies. And we did look at the variation of the concentration between 0.6 and 1, and we did work with cleaning procedures of about 30 minutes, which is probably longer than what you can find in the food industry, in order to see what happens if you continue the cleaning process. Okay, so what spores and what strains we did work with? We decided to work with 4 strains from dairy processing lines, and two Bacillus cereus strains and two other species, Bacillus pumilus and Bacillus subtilis. Cereus and the two cereus could be considered as potential pathogenic bacteria, and you can correct me, not for the others, but the others are potentially present in milk and could be involved in the shelf life, reduction of the shelf life of the milk. So through this transmission electron microscopy observation, it's possible to, I don't know if you see something, even with my glasses I have difficulties. Here it's, I just want to show you that here you see the spores. I don't know, no, it won't change anything. You can trust me. Here you have a kind of membranes, which is considered to be the rest of the vegetative cells. And the name of that is exosporium. And here you have, you can see quite large, huge exosporia. It's the same for this strain, but for these two other strains, there is no exosporia. But it's possible to see appendages, to see different elements on the exterior of the cells that could be of help in explaining what happened. So to test or to evaluate the contamination through the cleaning process, what we do, we do two different techniques, which are not really very I don't know how to say. It's not it's not based on molecular biology and so on. It's just basic methods by direct culture on the surface. So it's possible to have the growth of the from one spore to a colony, and this colony is possible to be seen directly on the medium on the nutrient medium by using a compound in the nutrient medium, which is TTC, and the colony appeared red. So it's possible to count the red spots you have on the surface. So you, for example, you have your pipes. You pour in your pipes your media. This is media that, in fact, you have a mold. You have and you remove this mold after letting the nutrient media on the surface. You have the growth of the bacteria, the remaining bacteria on the surface, so you have a colony. When you remove your nutrient media, which is like a cake, you can see the different spots corresponding to colonies, and you can count it. So it's good when you have a very low amount of bacteria remaining on your surface, which is about 10 to 100 per square centimeters, which is impossible to evaluate by using classical technique as swabbing and so on. So it's a direct evaluation of the contamination on the surface. Under the microscope, it's possible to see the cells and to count the cells, and by counting the cells to evaluate how much are still remaining on the surface. So it's very simple. I don't want to detail more because it's quite obvious. For the model used, we didn't look at a very elaborate model because you will see the shape of the curves, of the kinetics curves, and we don't want to have a model with lots of parameters that are very difficult to explain. And here, these parameters this model was used for the removal soil from a plate heat exchanger for a tubular heat exchanger, and it was proposed by German team in the 2002, and so we found it quite convenient to describe the curves we have. And these two parameters are, the first one is delta parameters. It's a time necessary to reduce the population by 1 log, 1 power of 10, which is quite simple to see, to understand. And the other one, P, is related to, could be related to the interaction between soil and surface within environmental conditions, the specific environmental conditions of your experiments. And if P is under 1, it means the longer the cleaning time, the more difficult it is to remove the soil. You have some situation where the longer the cleaning time, the easier it is to remove the remaining soil. It's important to know that, because if at any time, if the remaining soil is more and more difficult to remove, it means that you will have a plateau and contamination remaining forever with your cleaning conditions. So this is based on this very simple model. So for the action of the sodium hydroxide, what was found is that the reduction of the population of the bacteria on the surface for the 2 Bacillus cereus strains, this is something which we did expect. There is a diminution of the remaining amount of bacteria with increase of the concentration of the sodium hydroxide. Nothing new. But for the two other strains without exosporium, what we found is something that we were surprised of, is that the diminution of the remaining population appeared to be very effective when you use a quite low concentration of sodium hydroxide. But when you increase the sodium hydroxide concentration, the diminution is not better. You don't improve the effect on the removal of your bacteria. And there was in the lab some observation on this, for example, on the layer, the outside layer of Bacillus subtilis spores, sorry. And what we found, that the thick layer surrounding the Bacillus subtilis spores was removed. And there were external layer of the spores that could be seen to be affected. So, in fact, there is an effect of the sodium hydroxide, but this effect will induce, it appeared to be induced, the attachment, or the change won't, the bacteria changed by this sodium hydroxide will remain on the surface. If we combine chemical action with a flow, so mechanical action when you clean, in cleaning in place, of course, there is a steam for Bacillus cereus. You can see here a diminution of the population. So you have about 3 log of 3 power of 10 of diminution of your bacteria population on the surface. However, for Bacillus subtilis and Bacillus pumilus, it's the same as the just chemical action. The bacteria appear to remain on the surface. But what was more interesting to see is that the fact that when you try to remove these spores from the surface by only mechanical action, it was only seen on this Bacillus cereus strain. And my colleague Christine Fay, microbiologist in the lab, she did work with a group, And she tried to remove all these spores, not only this one, but different, maybe about 10 to 20 different strains of different species. And what she found is when in the food industry, when you clean processing lines, the forces you have on the surface when you clean it, you can use this in order to have a better idea. When you clean normal cleaning, you have wall shear stress. I won't go into detail about wall shear stress, about 2 to 10 pascals. Okay. And it's directly related to the force, the force at the surface, the force able to remove. Okay, and we did some experiments by working not with 10 pascal, but with 700 pascals, which is impossible to have in the food industry when you clean. It means that you have pumps that with a range, impossible. All your processing line will explode. You see, and the spores were not removed. So it means that the chemical action is important. Of course, you need a chemical action, and it's not obvious that the combination with between chemical action and mechanical action is that effective, and we've seen that on these two strains of Bacillus, one of Bacillus pumilus and one of Bacillus subtilis. Fortunately, it's not for Thiery, which is a potential pathogenic bacteria. If you look at the parameters, because I did some explanation about some detail, I went into some details about the model, and if you look at the time necessary to remove 1 power of 10 for Bacillus pumilus, you need 1 hour 45 minutes. If it's Bacillus subtilis, it's 4 hours. For Bacillus cereus, it's 6 minutes and 5 minutes for these two strains. But by mechanical action we use in our experiments, it's 1 day. And you count in days to remove. It's no sense, but it's just to show the interest of this kind of parameters. And you see that all the P parameter, all of them are under 1. So it means that the longer the cleaning time, the more difficult, the less easier is to remove the remaining bacteria on the surface. Some related issues. It's not finished. We did work on Bacillus biofilms. And for example, the growth of Bacillus, here it's Bacillus cereus, the easiest one to remove. What we found is when you have a non-complete coverage after 48 hours growth on your surface, but what was more interesting is that the fact that in the biofilms we found between 15 to 100% of cells which are spores. So and you can tell me that it's because 48 hours. What means 48 hours in the food industry? And we start some work to see at what time, what is the lag time necessary to found spores in the biofilm. And at the moment we have seen that the spores are formed very early in the growth of the biofilm. So but the other question is how these spores are could be compared to the spores we worked with in the other experiments, because when we did work with spores, it means that you prepare the spores, you grow your bacteria in a spatial media to obtain to have the more spores you can have on this spatial media, and you collect all the cells. You kill the vegetative cells, you clean the spores, and you use the spores for other experiments. You see what I mean? I don't know. I'm not sure to be clear, but you have to manipulate, to clean, and in order to have only spores. Here it's spores within a biofilm. It means that it's spores with an environment which is not clean, in bracket. Why? Because in biofilms you have exopolymers produced by the vegetative cells. So maybe these spores with the surface properties because of these exopolymers could be different, and the behavior could be different. So something that we have to look at. We did some experiments with these biofilms full of spores and look at the removal of the spores in the biofilms. And what we found that is, for example, with the CIP cleaning, the same conditions as I mentioned in the first experiment, for the first data, we found that the diminution of the reduction of the population is quite rapid. In less than 3 minutes, you have 3 log reduction. So it's this is the explanation could be the fact that the spores are different because the surface properties of the spores are different, or probably these spores are not directly attached to the surface, but are in 3D structures in the biofilm, which is easier to remove than spores just attached on the surface. This could be so. The result of that is in 3 minutes you have the 3 to 4 log reduction. But what was more interesting is to see that after that, no removal was observed. No more removal was observed. So this is to my mind, this is an issue. Why these spores are still on the surface and still difficult to remove. And the last figure I will want to show you is some experiment done in the lab 2 years ago, 2 to 3 years ago. Here it's some strains of Bacillus cereus, and the one you can see in every experiment, 98/4. And we decided to soil a pipe with a huge amount of spores and to integrate that in a clean line, processing line, and have a look on re-adhesion, potential re-adhesion, if there are re-adhesion of spores on the rest of the clean processing line. When cleaning, not by just rinsing, you have demonstrated, you can trust me, that by rinsing you don't detach anything. So it's cleaning. And what we found that, first of all, that there is a re-adhesion when cleaning of spores on the pipes along the lines. And the other things we found that here it's for pipes, that just after a bend, the organization of the flow will induce higher attachment on the surface. And we went further than that by looking at the recontamination of a valve, for example. And we found that the recontamination was related to the geometry of the valve. And it was possible to find with this technique, with mold, agar media mold, to see where were the poorer hygienic areas in this valve. And in order to it was possible to relate the re-adhesion to the organization of the flow inside the valve. So spores are in the flow. They can be trapped by the surface, and the trap phenomenon is induced, is favored by the geometries. So, and this is something that could be important for food producers. That readhesion is something that could happen when cleaning. So let's go to the conclusions. So high adhesion forces, which is something not new, but what was new is the fact that there is a really high resistance to mechanical action. Chemical action clearly is clearly effective, so it's good for detergent producers, but it's not sufficient to have just chemical action. And and something that could be we did look at is the action of sodium hydroxide. It's not really a detergent, but it's widely used as in cleaning in place conditions in the food industry. And so there is a real action, obvious action on the wall of, for example, Bacillus subtilis spores. But the results of that is it seems to enforce the adhesion of the spores on the surface. So there could be a high risk. I don't know if I can say high risk. A risk of food cross-contamination in processing line. And the last issue is the fact that in Bacillus biofilms you can have up to 100% of spores, and these spores are able to recontaminate even when cleaning. So we still work on mechanisms involved in the removal of spores and of course of biofilms, and it's fully related to how to lower sanitation environmental impacts. So the question is, of course, we see it's possible to see that there are some difficulties that could explain some presence of these bacteria in food, these spores in food after processing. But for the future, the next target is how to reduce the environmental impact of the cleaning conditions and all sanitation conditions already used in the food industry. Because the question is still, why using 2% of sodium hydroxide and not 1%? Is it more effective or not? Some data here show that it's not more effective, except for Bacillus cereus, but the answer is not that obvious, you see. And the other question is about the length of the cleaning process, the cleaning time, if after 10 minutes or 5 minutes there is no more effect, what to do? So the possibility, one possibility is to change some conditions, and we try, for example, to use pulse flow. You see. So with pulse flow, you change the reorganization, you force the reorganization of the flow, so you change the orientation of the forces. And what we found is a better elimination of some spores. Some spores from some strains, sorry. You see? So we have to re-engineer the cleaning process in order to not to say that's it and it's not possible to be better. Of course we can be better. but we have to think about all the technologies that could be available for this question. And for all these techniques, what are the environmental impact and how to minimize the environmental impact and how to increase the food safety or maintain food safety, because food safety is not that bad in our countries. Okay. I'm sorry to be too long.